to where she could give it a scare. Who knows?
And then
And
shot it off?
shot it off. Yep.
Interesting.
Any chance there was any nutritional value on any material that might be on the shell?
Well, you know, they have a beak that they pick their food apart on, and there's not, as far as I know, there's not digestion outside of the body like you have in echinoderms. So I don't know. It's hard to imagine that happening.
So we're measuring these persistent organic pollutants in the water. Are we doing any sediment tests? Are we collecting? We are.
Yeah, so we are on the lookout for a nice area where we could take 1 or 2 sediment cores, but the ROV has to be able to settle down, and we need to see flat what seems to be sediment maybe a few inches deep. So we are going to be measuring these same contaminants in sediments.
Gotcha. If I missed this before, I'm sorry. Do we think there's a higher concentration in the water or the sediment?
Probably in the sediment.
Probably in the sediment.
That's usually the way it goes because, as I said, these things tend to stick to sediment particles and they settle out and wind up in the ocean. But it's hard to, I mean, we measure them in different ways. One is by weight. So the weight of contaminant per weight of sediment, and in the water, it's the weight of contaminant per volume of water. So it's hard to be equivalent, to equate them. So part of what we want to do is come up, see what the equilibrium factors are, so concentration in sediments versus concentration in the water. And that varies among the different kinds of contaminants. And one of the reasons that we're really interested about these, not only are they periodically at quite high concentrations in Monterey Bay, which are, of course, directly of interest by Monterey Bay National Sanctuary and the effects that they could be having on resources in the sanctuary, but we know that one of the factors listed by NOAA as impeding the recovery of the endangered southern resident killer whale population along the west coast of the U.S. is high concentrations of these same persistent organic pollutants. So they know that the concentrations of these things in the blubber of killer whales are higher than the concentrations that were found in the blubber of harbor seals in captive feeding studies. And those concentrations in the blubber of the harbor seals were found to be related to harmful effects on their endocrine system and their immune system.
So they're getting that from something they're eating?
Yes, these things biomagnify up the food web. They tend to be soluble in lipids, so they've done these studies by sidling up to a killer whale, hitting it with a small harpoon that takes a plug of blubber out, and then they analyze that blubber. So yes, it's in the food that they eat. In fact, they use the profiles, the relative concentrations of different kinds of contaminants, say DDTs versus PCBs, to kind of determine different feeding strategies in the killer whales. So some populations, some that feed in California, have what's known as a California signature, where there's high DDT concentrations relative to PCB concentrations. So, and that's because the California population tends to feed on salmon, which are very fatty fish, and there's a lot of DDT coming off the agricultural watersheds in California still. and that's contributing to the higher concentrations of DDTs in the killer whales that feed off the coast of California.
Wow. So these chemicals can really impact our top predators as well as the bottom.
Especially the top predators,
Especially
because
the top
they
predators.
bioaccumulate and biomagnify. So,
would you kindly like to describe biomagnification or amplification for us?
sure. Biomagnification is where, with each kind of step up the food ladder, food chain, concentrations increase. And that's what we typically see in contaminants that are lipid soluble. So, and rough rule of thumb is a 10-fold increase up each step of the prey ladder. So at the very top of the food web, you find the highest concentrations of these contaminants. So, you know, bigger animals eat more more volume, more weight of prey than smaller animals, and so they're accumulating all the persistent organic pollutants from the prey that they eat. So as they eat more, they accumulate more, and so the concentration goes higher and higher up the food web.
Builds up and builds up. Gotcha. There is a ground maintenance industry worker, and they are asking, which chemical do you believe is the worst? What do we put down that ends up causing the most damage?
Well, that's a really good question, because there's all kinds of emerging contaminants that we're beginning to find are causing problems. So new ones are coming up all the time.
Oh no.
I think that the biggest problem is with contaminants that are no longer used. These things that don't break down in the environment, they tend to be chlorinated, and they biomagnify. So even though, like with the case of PCBs and DDTs, their use has declined steadily since the 1970s. In fact, in the U.S., DDTs have not been used since the 1970s, but still it's washing off the land. They stick to
Looks
sediment.
like the end of the line to me.
So,
is it open sediment below you?
Nothing but mud.
It could be.
Can we get a sediment core? That would be great.
We can try.
That's
The
definite
vehicles are
sediment
120
core.
meters behind us, so
What do
we
you
can stop
recommend
here
so-
and hope that it's still sediment when we stop.
What do you recommend in terms of being able to start the next transect with the best turnaround time? Would taking a sediment core inhibit that or delay that, or not really, and we can try it?
It would slow us down a little bit, but sediment core is probably 10 minutes once we come to a stop. I don't know.
And for the ship moving on to that next line, is that going to have to wait, or?
Yeah, like once we stop the ship, we got to cover maybe 80 meters before I'll start moving to the northwest. And then we'll wait for Argus to sort of catch up again and then start to the southwest.
The ship's at the end of the line there, isn't it?
Yeah, more or less.
So if you stop her now, we can swing in and still get one.
We should try and get one.
Yeah, we'll stop the
agree.
ship now. It's at the end of the line.
Yeah,
We'll get set up
this
for
looks
a
like a great spot.
See if you can give me something to look at while you're doing your push.
Put on a
course.
movie.
Just a feature, anything like this.
Like this octopus here?
Like
Is
this
an
octopus
octopus?
here would
How
be
about this
perfect.
octopus?
We
Perfect.
can tell you some fun octopus facts. So we learned before that they have 3 hearts. That's correct?
Yes, that's right.
They have two hearts around
We
sort
are.
of their
We're going to
gills,
continue online.
and then one to pump
3.
blood to their body. We learned that, oh, he's getting a little dusting.
So let's turn the pump off now, momentarily. Is it off now, Dan?
Not
still
yet.
flying.
Okay.
I forget where we plugged it in. The pump is off.
Okay, thank you.
PCB pump is off.
Okay, we're going to say that's 0224.
Yeah.
Yeah.
Whatever one you like. You can 5. Choice of 5 there. I can do the sample tray for you whenever you're ready.
So the ROV is going to pull out a push core and shove it into the bottom and pull it up, put it back in the rack. And hopefully it comes up with a nice plug of sediment.
You want one of the little ones on the big screen there?
I'm not. Oh.
Which one?
Oh, yeah. Either one. Any one that's easy to get to. Yeah, perfect.
Sorry, I stuck it in my co-pilot.
Okay, got it. Okay.
We've got an octopus physiology question. Can an octopus live without one of its hearts? I don't really think it could.
Well, if it doesn't move much, maybe it could do without one of those branchial hearts.
Yeah.
So, but the hearts are for pumping. 2 of the hearts are branchial hearts, which pump blood across the gills, called tinidia, and one of the hearts is a systemic heart, which pumps blood through the rest of its body. So I think it definitely needs that systemic heart. I suppose if it was not using a lot of energy and so didn't need to respire as much,
You wanna
maybe it
come
could
down
get
5
away with
meters?
1 gill. But not nearly as well as with all 3 working.
Yes.
Yeah.
That's an
Okay.
interesting thought. So I know a fun fact about squids is I believe that their brain is kind of around their esophagus or their mouth where their food comes in. Is that the same sort of thing for octopus?
1 meter.
It is. And in fact, that's a really sort of a shared ancestral feature of many invertebrate groups.
That's
So,
2. That's 3.
Octopuses are mollusks, so all mollusks
4.
and arguably all
That's
protostome
5.
groups of mollusks or
Okay.
of animals have nerves that wrap around the esophagus, which in some become ganglia, which is basically a thickening of nerves, which is the beginnings of a brain. So yeah, there are a lot of things with nervous ganglia wrapped around their esophagi. It's pretty neat.
That is very interesting.
Yeah.
Seems kind of challenging though, right? Because they got to break down their food into pieces that will fit around
Between,
their brain.
I guess. So works for them.
Through, yeah, through their brain. You swallow through your brain.
That's true, but if you ever start reading about how air has to move through from the blowhole of a whale to its lungs, you'll realize we've got some key problems in our chordate or vertebrate morphology too.
Oh, I've never thought about
So,
that.
yeah, dolphins have a bifurcated is it esophagus. So it means that the trachea, the airway, goes straight through the esophagus, and the esophagus has to branch around it, which means that if a dolphin
Wow.
swallows a fish just right, it can, well, starve to death because it gets stuck there at that branching and then can't eat other food. But it could still breathe.
But it can still breathe. Wow.
And then there's the opposite problem of baleen whales. They have some sort of little flap that has to go up or down to direct food and air to the right places. So it's something like the trachea and the esophagus both at one point cross each other, which is very similar to actually what we have. That's why we can swallow wrong and end up with food or liquids going down our airway.
Who knew it could be so tricky?
Who knew? It would be so much better if you were like a sea cucumber and you had a cloaca, so you could just breathe through your cloaca.
See, never knew how useful a cloaca could be.
See?
Look at the sea cucumbers. They've got it all figured out.
It's a pretty good life.
Let's see. Do other octopus produce venom like the blue-ringed octopus? If so, how common is it across octopoda? If not, why do you think the blue-ringed octopus developed it?
I know that other octopuses do produce various types or strengths of venom. Not any as potent as the blue ring octopus, but definitely many octopuses will use their beaks to bite, but then also envenomate their prey with the radula structure behind it.
Yeah, that sounds kind of scary. But
Yeah.
You
I
guys are gonna get.
reading a book in
Oh, you're
regards to
driving along.
humans, and we know a little bit about octopus venom, but not a whole lot. So different people react differently
We're
to
going to bring it around in the front camera to stand by.
venom. So we're kind of unsure of how venom can interact with us as humans.
There was a popular news article maybe a month or two ago about someone who
Oh, that lady.
put an octopus on her face and then it bit her
Yes.
and her face went paralyzed for a while. So we can be affected by the venoms.
Absolutely. We can definitely be affected by them. What effects we have definitely seem to vary. So I would recommend not putting an octopus on your face.
Just leave them alone.
Leave them be. They're very smart creatures. And you would be startled too if somebody just picked you up out off your couch and put you on their face. So
Here we are with the push core.
oh, here we go. Let's get some sediment. So we're testing for
push core number 1.
these persistent
Yep.
organic pollutants. Are we
Correct.
looking for anything else in the sediment? Are we going to try to look at what sort of geology we have here?
No, although we don't need all this sediment.
Okay.
So if people wanted some, certainly we could save that. I don't know.
Anyone need some science? We're here to provide. So all of the flakes and the sediments and the things that you see moving around are some of the sediment that we
Gotcha.
have kicked up from settling down here before getting our core. There is also what we call marine snow, so that's sort of different. Oh, there goes a little red jelly. Different matter falling down from above, whether it's sloughed off skin cells or maybe some fecal pellets or bits and pieces of dead animals or dead plankton. All of this stuff is falling down slowly to the bottom of the ocean and can provide a lot of nutrients for the things that live down here.
So Hercules, I think the farther from the edge of the rocks we can get, the deeper the sediment will be.
Yeah, copy that. If I was going to try and use the corner of the rock here to perch, because if we
Okay,
set the
got it.
vehicle in that mud, you'll be looking at it for the next hour.
Yep.
Because it does a cookie cutter, but I'm really tugging on Argus, so I don't know if that's possible.
Okay.
I'm going to try. Can you do bubble 1? She can also swing the manip out. So if you're happy with that in there somewhere.
Yes. Yeah. Yeah.
Roger. I can land out in the mud if you want, but I might anyways. I'm now pulling Argus and 3,000 meters of cable with a
Wow.
20 horse
Okay.
vehicle there.
Yeah.
Witness the full power of Hercules. Lift your arm up a bit. Good. Lift up a bit more. Up, up, up. Sorry. So you're going to have to move that
Shake
off
it
to the
out.
camera and
Yeah.
shake it out. Shake it, shake it, shake it.
Shake, shake, shake.
Outside
Move
the
out.
camera range.
Yeah, out to the right.
Lift
Sorry,
it up.
I stuffed you.
That's okay.
Didn't
Not
look
that far
deep
out.
here.
Not that far out. Bring it back just into the camera. There you go, just so you can see what you're doing.
Well, that's really.
Ah, it flew out.
There it goes.
Shift.
Yeah, keep going.
Left and right, left and right.
Yeah, move your wrists back and forth like you're dancing. There you go.
Okay, but.
Okay, so
yeah,
now you want to bring
what
it
we.
back to the.
I mean, sediment is sediment. So.
Set to the left of that hole, and try and get it perpendicular to the soil.
Ooh, you're in the light bar.
Up.
Shake again. So it's really steep here, so we're hitting the light bar. The bonus is it's going to be really sticky, so you want to keep doing that wag till the tube's clear.
Would it be easier to reach and to get that
Okay.
if it was to the right? Like the right
So take your time now. You got all the time in the world, and you'll want to bring the elbow out.
And way to the left of that disturbed spot.
Yeah. Might be too steep here to get it at that angle.
What about on the right-hand side of the screen, Dean?
Yeah, that's fine too.
So
Seems
bring
easier
your
to
pitch
reach.
all the way up. You don't make sure you don't get it in the dirt
yeah.
again. Give her a shake again there. Okay. And now if you keep it turned up and then bring it back. There you go. Now bring your minute. Turn it back up out of the, so we don't stuff it in the mud again. There you go. Now bring your arm in. So move nice and slow now, nice and slow.
So,
if you hit the light bar of the mud, you're too close. And the wrist is proportional too, so you can like barely, so you got a little up there.
Yeah,
I would go right up
lift
to where
it
you're
up.
almost touching the light bar. There you go. Super close. And a little more spin on that wrist, but touch it real easy. It will spin slow. A little more maybe. A little more, a little more. That's looking good.
Yep.
You happy with that?
Yeah, I am.
Okay, so nice and easy, steady push. And you want to try and not move it around. You want to push straight down. Beautiful.
All the
Keep
way you
going.
can.
Keep going. Yeah,
Let's
keep
rock.
going. You might lift the vehicle up a little, but give her.
Still going.
Yep, still going.
All right, push a little harder. And be ready, it might break through. And that's probably deep enough there. You happy
yeah,
with that?
Definitely. That's
good.
beautiful.
Okay, now up nice and easy. Now you want to move slow the whole time because you don't want to shake the sample out.
Taylor in, this is sample number 17.
Yep, that's what I have.
It's not in the box yet.
Yeah, right, okay. I won't log the time.
It's easier to delete than to create.
Right, you get it?
That's it. So it looks like we got a nice
Half.
sample in there.
Yep.
So watch the light bar, and then let me come up a bit. I'll get out of the mud here for you. Okay, beautifully done. Now you should be able to swing around easy, and the hard part is putting it back in the holster. That was the easy part. Watch the light bar
Yep.
there, Hunt. Come down a little and then
Slow
swing her right.
movements.
Yeah, nice and slow.
I haven't been
And if
taking
you
notes
remember
here.
where the arm was as you're swinging
Oops.
around, you can get ready so
Oh, I
you'll
did
kind
have
of be
to
tucked
put
in.
the time off, but I didn't have any time
I can
ons.
drive bubble for you if
So
you
here's
want.
the off time.
I'm gonna try like 223.
Oh,
But
224.
basically you gotta come around till you can see it in the other cameras. I'm gonna give you loud and proud on the big screens, all right?
What transect was that? That was,
That was 910.
okay.
We've got a question of what kind of time scale does that core represent? How long
Yeah.
did it take to make that layer of sediment? Do we have any estimate?
No, we don't.
We do
have,
not.
yeah, that's a really good question. Yeah, have no idea.
Do you think we'll be able to see some sort
So
of
when you
different
do a wrist twitch,
geological
just do it
layering?
super slow. 'Cause
Well,
it's really
we
touchy.
might be able to see different layering, but
No, you
not
pretty much gotta get it
necessarily
lined up
any
straight up and
sense
down.
of the geological time frame incorporated in them. So the layering would probably be like well oxygenated versus
You're
oxygen
getting closer.
poor. So, and at that point where it goes from oxygenated to
All you gotta do is get it in about that far, and then you can
non-oxygenated.
let go of it.
yeah, the sediment turns black. So it looks like it's a, I don't know, pretty consistent color there, but we'll know more once we get it on board.
All right.
In young sediment layers like these, the way you can determine how old a core that thick would be is to put out what's essentially a rain catcher. That's called a sediment trap
Yep,
fill. And so it would sit out with an opening at the top, and
Look,
sediment would fall
concentration.
into it over time. And so you can look at how much sediment has accumulated over some known amount of
You're
time.
really close.
But the difference is that sediment traps work because
So
the
it'll
sediment
be,
particles settle into
Elbow up and pitch down a little.
a tube where the water is quiet and no longer able to keep the sediment particle in suspension. So I don't know that they know direct correlations. I don't know that, well, some people may have done quantitative studies where they know actual sedimentation rate on the seafloor versus what's captured.
What'll
in
help
the
you a
sediment
little
trap,
is if
but
you swing
I
your
don't know that.
azimuth left a little and
Yeah.
yaw to the right a little.
There are a couple of different sediment trap configurations
Yeah,
that
like
people
that,
have
and
used
then
to try to
elbow up.
get at those different mechanisms.
mhm.
So some sediment traps will be tubes that stick into the seafloor, and they have holes on the sides, so they would capture sediment that was moving laterally.
Aha. So they're capped on the top, essentially.
That's
Yes.
good.
Aha.
Now use your wrist.
Or they could
a
be
little.
open if you want both types.
Just a little rotate on your wrist, and I'll get you where you want to go. Yeah, a little more swing will get you. Just swing right and you have it. Once you get it lined up with the other ones, you should be able to use your wrist to twitch it. I'll stop talking now.
We're collecting our sediment sample. We're putting it back into its container. But
you'll want to yaw
very
right.
tricky,
Yeah,
you
yeah, yeah.
guys.
Easy,
Sort
easy.
of
So you
seeing
can see layers in that
2D,
sediment.
but you're working in
Yeah,
3D.
you're right.
Yeah, azimuth left, yaw right. think about this part of it. So, so you have to swing the azimuth to the left a little. The big guy, yeah, azimuth left. You're frozen there. There you go. And then bring your yaw, this guy, to the right a little. And then wrists rotate clockwise.
I've got
There
a
you
good
go.
question here.
And
Do
then elbow
our
out just a little.
POPs include microplastics? Are we including
Helps
that
me
in
to
our
picture the
study?
arm in my head. Which
We
way I
are
want
not,
it to move.
but
So,
what is really interesting about microplastics is that they tend to be chemically such that these POPs stick to them. So
Uncle? Uncle?
not only
Hey, I'll get you lined up.
not only are microplastics a bad thing, but
That's all right.
animals
Hey, I'll
that
get you
wind
lined
up
up, and
ingesting
then I'll
them
let you put it in.
get a goodly dose of POPs.
You did. Oh, you're there.
Slow, 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 slow.
They're really close.
Yep.
This is probably one of the hardest things we do is
Yep.
getting these cores back in.
It is a tricky way. You're saying before it's hard to see what you're doing, right? Because you're looking at a 3D environment in 2D.
We're looking so that we can't see the arm, and it's backwards in the camera. So the move you think you're doing is
The opposite of what you think.
180 out. Yep.
We're moving in opposites here. There we go.
Nice work.
Well done.
Thanks for the college try, Antonella. We were sweating with you.
I'm having trouble getting on the ball to give it the love tap. So, come on.
How long is this arm of Hercules? How long is our predator arm?
The reach is about 2 meters.
About 2 meters.
So we give it the love tap there to seal the bung in the bottom.
There we go.
Okay, take this thing back and park it, please.
Science collected. So that's going to tell us a lot about the sediment, the different pollutants inside the sediment. Maybe if we get a...
Is it just the one we want here, or do you want...
Yeah, no, that one...
Line number 3.
Yeah.
Thanks, guys. That was a long time coming. We were
It was.
praying to get a sediment core, so
Yep,
thank you.
yep. We
Thank you for your
appreciate it.
patience.
No,
No problem.
absolutely. Thanks for your help.
Yeah, so
Okay,
next up
salvo
is to head
norma.
back to that line 3. And Dane, you let us know when you want the pump turned back on.
Yeah, so it's pretty clear here. If we could get the
Hey,
bubble
look, we're still
cam
here.
looking at that pump inlet, or pump exhaust. I want to see how those telltales lie.
So we're ready to head to the next line, are we?
Well, before we do that, can you turn the bubble cam so that we're looking at the pump exhaust?
Yeah, I can do.
Oh, we can start heading toward that. It's going to
Well,
be a while
wait,
to get the
before
ship
yeah,
moving and
before we
everything.
move. Okay, now tap the pump on.
The pump is off, I believe.
Yeah, tap it on.
Oh, Roger.
wanna
Let's see if I can
see-
find it. Let's see, where did we put it? We put it here under science. PCV pump is on.
It's on, eh?
In theory, yeah.
Okay.
And Dane,
Yes.
is this the third time you're turning this on?
Yes.
time?
Let's leave it on.
Okay.
Yep.
All right. So I'll put this in the book, and then once you turn it off again, then we'll record that as another sample.
Right.
Okay, cool. Sounds great. Start time for that is 0248.
Okay.
Can you tell us a little bit more about what this pump is collecting?
It is sucking water through the sample media that will be taken into the laboratory and analyzed for persistent organic pollutants.
So this is pulling that from the water,
Yes.
and whereas we just did a sediment grab. So we've got both of those.
Correct.
Awesome. You said it was going on to a media? What kind of
It's a
media
benzophenone.
do you
Well,
have?
so there's a stainless steel column that is filled with these little tiny sub-millimeter sized
Dan,
resin
can we give him a little bit
beads.
of internet famous
And
time?
the persistent organic pollutants stick to those resin
The
beads.
little Meduse. Let's give him some internet famous
and
time.
don't
Yeah, that sounds good.
come off. And so in the
It's a
laboratory,
benzocodone.
they get extracted with solvents and then measured.
Very cool. Lots of chemistry going on there.
Yes.
So you're chemists, you can also help out with our ocean exploration. This little red Medusa is adorable.
And
So
it is
shiny.
touching the lens.
He's touching the lens. He says high five.
Wow.
Very cool. Amanda, did you say the scientific name of this guy?
Yeah, Erica recognized it.
Erica.
Benthocodone.
One more time?
Benthocodone.
Very cool.
It's a jelly that you mainly find near the seafloor.
Yes. Sometimes in numerous quantities.
Oh, so he might be a little lonely.
I noticed on the Greater Fairlands and Cordell Bank, you saw a lot of these guys.
Benzopodone, is that like bottom foot? Deep foot? Oh,
Not
codon.
podon,
Okay.
yeah, codon.
Coming full wide. Which,
Off he goes.
It's staying on.
Is it running, Dane?
Well, it doesn't look like it's putting out that much.
Can we let them
there was
proceed to the
another
transect
little
line now,
one.
Dane?
Yes,
Or held up?
absolutely. Yeah.
Okay.
Argus Zeus is full wide at
Was
the
that
moment.
a go for ship move?
And then I can bump in to get rid of the vignetting.
So we're heading to 3 now. Line 3.
Yeah, that's the
Yes.
question.
Yes.
Am I allowed to move?
And I
Yes.
can
Yes,
Roger.
please.
drop the iris boost. This is where I normally like to leave it. I bump it up when you're in search mode to help you find it. Oh, you want more zoom? Okay. Yeah. If we can. Oh, sorry about that. Can. Yeah. Oh, well, hello. You're early.
Do we notice a difference between deep sea jellies and more tropical shallow jellies? One thing I've definitely noticed is the color. So the color red doesn't make it, the red light waves don't make it all the way down here. So lots of things like to try to blend in by being that red because it's much harder to pick up that color deep down. I think our scientists are having a
switch over.
consortium and a switchover.
The science huddle.
A science huddle, that's it. They're figuring out all our science. trying to answer some questions.
So,
Prep the next group as we continue with our dive.
yeah,
Yeah, we're
red.
passing the torch on to the next watch, so just sharing what we've done so they can carry on and we know what's happening next.
So we have got a number of different researchers here on the boat. We have engineers, our pilots, we have video, we've got our scientists. I'm your communication fellow. So you have all different ways to explore the ocean if you have an interest in any of those careers. Also, we are super excited that you guys here at Nautilus Live are watching with us and connecting with us. We're exploring Davidson Seamount at the same time that you guys are watching it. So we're working together here to explore an area that's never really been studied. So we're finding all sorts of new things. We're understanding what it is we're seeing, some of it if we can, and trying to see what else we don't know here about the ocean floor, the different creatures. We found some amazing octopus gardens, spas, shimmers. Beautiful stalked sponge. I have learned so much about sponges in just a few hours. This is fantastic. As well as the bony fish. We had a cutlass eel and a grenadier. Very interesting to learn the difference between those. There's all sorts of guys hiding out here, and what we're seeing right now, I see some sea stars, I see a sponge, there's an anemone, a crinoid. I'm gonna go with that's a very long coral, though I'm not gonna tell you which one because I think I'll be wrong, but it's beautiful there. So there's so many different organisms that are living in this environment. So I said red light doesn't reach down this far. Absolutely, I can explain that idea. So most light does not reach down this far, but just a little bit does. But those wavelengths are going to be our sort of darker blue and purple wavelengths. Our red and our orange are the first wavelengths that are sort of bounced out or not able to continue through the water. And so creatures down here that are red make it harder for them to see because you just can't pick up those wavelengths, so they blend in. What a great question. If the animals down there are nearly invisible, how are they able to find other organisms of the same species for mating purposes? That is absolutely a challenge for a lot of our deep sea organisms, and each of our deep sea organisms are going to handle it differently. So things like our anglerfish, which are very interesting. Once a male
Video, can we come
finds
full wide on Argus?
a female, he actually attaches to
Thanks.
her and becomes part of her body for the rest of the life. So once you find one, you're set for a while. Our octopus, as we were talking about before, have kind of an ability to smell and sense what's going on around them with their arms. So they may be able to smell and kind of feel and sense if there is a possible partner around.
Were you allowed to zoom, or what are we allowed to do?
So there definitely are multiple
Aaron,
ways that
go
these
for it.
species are trying to find each other. It may be light signals, bioluminescence. It may be smell. I don't know if sound is one, but it might be. Sound might give you away. But there are multiple different ways that animals try to face this challenge of, yes, we can't see, but we still have to find a mate.
All right, full wide. Yeah. Oh, Dan did get us a little dirty. Yeah, that's just probably a rust, and then it's paid out past that and then twisted a little bit.
All right, guys, this is Danny signing off. Danielle, thank you guys for all of your questions. I will see you, well, bright and early my time. I need a nap, but you'll have lots of fun with Megan.
All right, I'm going to start coming around a little bit.
Hey everybody.
Hello.
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 Alright, looks like we got nav handover still happening, but pretty much everybody else
You
came
know, the
online.
predicted highest one was like 18. And that was for the gust.
Mmm. Do. How's that? Sing you a little song. Okay.
Okay, so should I come down a little bit?
I'm heading here to this.
Not yet, because you're still over stuff.
Okay.
This track line 3.
We're at 37.
Yeah, I'm going to come up the wall a little bit and look
Okay.
at stuff.
Yep. Okay.
Are we making our way to a transect?
Yes.
Yes, all right, I think we've
Oh, so you're erasing the sample, the PCB sample
we
numbers
switched
that I was just
everyone over
informed
here.
about? Okay.
Hi, back row.
Hello, front row.
It's been a while.
No, I've missed you all.
It's been about 8 hours.
About 8 hours.
Feels like.
Yeah.
Found
Seems
rock.
like just yesterday, Chad.
All right, well, I'll confirm with you what the notes say, but
Or
for
earlier
right
today,
now,
I
I'll
don't
change
know.
this one to 1.5. What are you going to do? Are you going to
label those later or not? No, I mean like the actual PCVs. Are you gonna end up giving them sample numbers? Yeah. I see
Yeah,
what
it's
you mean,
not. Yeah.
Okay, so and this push core 015. Okay. And now I'll go hunting for the other
Am I
one.
clear to move bubble cam?
You are. Yeah. Thanks. So Jamie.
Oh, let me kill porch light. Roman Hotel is off.
All right, the last one I
Closed
have
for the
before
night.
that
What was
is
the
sponge
on
slurp.
time, back on time, on and off time for the pump?
Here, let me say this on SPL. We had the original on at 15:49.
Correct.
And then an off at 22:15.
Correct.
And then another
On at 22:28.
Okay, yeah, I had that as 22:24, so that's incorrect.
Yeah.
So yeah, 22. Wait. Oh wait, no,
Shark 'em
she
up a little bit.
missed. She missed that time. So yeah, 22:28
now
was
we
the
can
next
come
on,
on.
and then we had an off at
Probably
02:24,
Yes.
Drop off
Okay.
this at some point.
Yes.
and that's
It's
what we have.
not really resolved
And
on
what
the
was
map.
back on?
It's
I don't
just a
know when the
general-
next on happened. Actually, I don't know if it is on again right
It
now.
is.
We're
right?
still going. Yes, it is on.
So did it get turned back on after the push core?
Yes.
So then that would have been close to, the push core ended at 02:46. So
Nav, do you have a?
I don't think he has one.
Okay.
Greg,
think-
was it already on when you did the watch
It was
change?
on when I showed up for watch.
Well,
Yeah.
then I think minus a few minutes, we could assume that it happened around 02:48, and we might be 1 or 2 minutes off, but that should be pretty close.
Okay.
The previous shift here did note a time off and time on time just recently.
Okay.
A time off of 0224.
Yep.
And on of 0239.
Aha. Okay.
That,
Thanks.
oh, okay. Maybe they turned it back on before they'd stowed the push core, but they'd finished taking it.
Okay. Well, okay. That's good for me.
Video
Thanks.
pushing.
Jamie,
Yes.
so the last sample before that 17, that's now 15, is 014, and that's a sponge slurp.
Yes, that's correct.
Okay, so I don't have any
Okay, so they
targets.
must not have dropped a target for those.
Yeah,
so we're
Miles
good.
said he did not.
We're good then
Okay.
on being caught up. Thank you.
Okay,
It's like-
I'll cut up there.
Great zoom on this Pacific Grenadier. It's going inverted.
So, science, as I understand it, so we're heading towards line 3.
Correct, it's line
And
3 on nav.
As from what I understand, we went southwest on line 4 from about where we were in the middle. Came down and then came up line 5. So now we're heading to line 3, which should be all freshly uncovered ground, but we did not cover about midway from line 4 to the northeast corner. Is that correct?
Correct, yeah.
Okay. Just double checking and make sure we're all set.
Yeah, once we finish this line, I know we may have, I came in just now on the shift change and realized they had asked to go to line 3. It might have been slightly more efficient to finish that last 2/3 of the previous line, but that's okay. We'll just do line 3 all the way down, and then we'll try to rejoin.
All right, let's head down.
Yeah, I think
The
actually
one in
this is
between.
probably best to just
Starting
go
to drop
3.
off the cliff.
Okay.
All right, I'm gonna do another step to get you guys all lined up on 3.
Yeah, because we'll spin around and face,
Yeah.
what, southwest?
Yeah, so I'm gonna do a 50-meter step at 345.
All right,
And
and you're
then you
clear to
should be
come around
ish
either way, so let's come around that way.
Yeah.
Bridge nav.
That way, and you can keep coming down.
You can type in that move you were typing in. 345. Yes, that's correct. Thank you. He's already on it. George is just so good.
George is a pro.
He's just already up there. typing away.
You're not coming down. You're just barely eking out. There you go. Multitasking.
Yeah.
It's good practice. Yep. No, I copied. I paid attention. I read between the lines, as it were.
Yep. Yeah, nothing too wally out here. It's all mounds.
All right, Rennie.
So you
So
should
this
be
is
able to
line,
keep coming
which one?
around.
I'm sorry.
Which line is this? Or which
we
are we
were-
trying to get back onto?
We are approaching line 3, which will be the 3rd transect. It's still one of the in the middle section over this this feature where we've been seeing the octopus.
Fantastic. Can we have a weather report, Rennie?
Yeah, winds have been between 15 and 20, and currents less than half a knot. Winds are coming from the northwest. Swell has kind of picked up over the course of the day. Still okay. Yeah, it's all of these forces are a bit higher than the model, but still within working limits.
Yeah.
And as our plan is to come up be on deck around noon tomorrow, just after our lunch.
Everything's
so.
supposed to die down before then, but
Yeah, we'll keep monitoring. Go ahead, George. I do. Okay, thank you.
I don't
I
have
was
George
hoping
on
for
my
something a
panel
little more
and I'm
professional than that, but
George is the greatest.
a professional opinion. I kind of like that not everyone can hear George, because then it just seems like I'm laughing. Yeah.
Rennie's just making up jokes in his head.
Yeah, this guy
George
that I
is just
call
his
on-
imaginary friend.
Yeah.
He
He is,
goes.
I don't know, I'll consult
George
George about
moves the
that.
ship. Yeah.
George. Yeah.
And what's our bearing for the line?
The bearing for the line, 213.
213.
Not
a degree more or less.
Exactly. All right, Paulo, you ready? I have a question for you right out the gate.
Okay.
All right. The question is, what's the difference in how you maneuver the two vehicles? So do you want to talk a little bit about what you were doing to maneuver Argus?
Yeah, so Argus has only 2 thrusters at the forward or the aft and rear. And so it's able to position side to side, or it's able to change its heading. But the height of Argus is controlled by a winch from the ship. And then we move the ship, that's what Rennie has been talking to George about, to move the ship to help position. And Argus will kind of swing through the water column because it's very heavy, even underwater. Whereas Hercules is slightly positively buoyant. And it's got a more flexible tether, and it's got 6 thrusters, so it can fully fly
Aaron,
kind of
let's
freely.
push in on the yellow thing. Yellow sponge.
I still expect there to be quite a bit of Argus movement, so I'll wait till we settle out before making this transit. Yeah, and the deeper the water, the longer it takes for that pendulum to swing. So patience has been
a virtue.
Virtue.
All right, full wide.
Rennie, do you feel like you
Let's
have a
turn our
good,
lasers back on.
do you calculate that? People often want to know, like, how long does it take for it to pendulum swing? Like, do you have a sense of it in your mind, or are you just kind of watching and responding?
Watching and responding, but we started to take measurements.
So it's come down some?
I think 2,500 meters, it's like 4 minutes before you even see Argus move. So we're a bit deeper, so.
Yeah,
5
a
minutes,
complete
like
move
that.
will take can take up to 15 minutes.
Yeah. And then you know, there's so much water column, so that last bit it takes a really long time to settle.
All right, Aaron, let's push in.
But it's almost immediate response, and if we're in a couple 100 meters of water, I mean, Argus, we know exactly where it is. It's dangling from